All right, good morning, everyone. 20 people here. Okay, that's all right. Whoops. All right. Yay, good morning. All right, we're gonna finish up the lecture material today. At least that's the goal. As next week we have our special guests coming. So good morning. Let me share the screen. Yes. All right, we're going to jump right into it because we got a decent amount to get through. Good morning, everyone's saying good morning. That's nice, thank you. Let's pull this big here. All right. Hopefully you guys enjoyed rocking to a little Aretha at some point. All right, so last time we kind of left off around here. I'm going to back up a little bit because we're talking about angiosperm reproduction, right? And as I said last time, you know, there's perfect flowers and there's imperfect flowers, and obviously reproduction happens in plants with either type of those, but for our simplicity, we're going to go through angiosperm reproduction using a perfect flower, just because it's a little bit more straightforward than dealing with two separate flowers. So I showed you last time we have the female reproductive structures, we have the male reproductive structures. We talked about the different parts of the female reproductive structures, right? Or collectively the pistil, or collectively the carpels, or collectively the gynosium, whichever one you prefer. Right, stigma style ovary. Inside of that ovary, there are one or more ovules, depending on what species you are talking about. Right, ovules. So ovules are where the female gametophyte and egg are going to be located. We've already learned about ovules in the other seed plants we've covered so far, right? The gymnosperms. Whoa, sorry about that. Spam call. So we learned about the ovules when we covered gymnosperms, and back then, when you were talking about the ovules, were back in the gymnosperms, were either in female strobili, like arranged in female strobili, or on fertile crown, leaves of a fertile crown, right? That was in the cycads. Or they were paired on a stalk in ginkgo, or they were on cones in the conifers, right? Those were in the gymnosperms. Now we're in the angiosperms, and the ovules are inside of flowers. Particularly, they're inside of the ovary of flowers. We talked about last time that because the ovules are now inside of an ovary and they have two integuments, they are more protected than the ovules that we saw in the gymnosperms. And then I made a big point of telling you, like, you know, the female gameto. So a real big concept of this course is how over evolutionary time, the gametophyte plants get smaller and smaller and smaller as plants evolve, right? And so when we get to the angiosperms, and we're talking about the female gametophyte plant, now we are only dealing with an 8-celled, or really 8-nucleide thing, which is the female gametophyte plant. So eight cell units basically is what the female gametophyte plant has been reduced to. And those units are 3 antipodal cells, 2 synergid cells, 2 polar nuclei, and then of course, 1 egg, right? Because you can't have an ovule without an egg. Alrighty. Oh yes, embryo sac. Okay. So today we're going to learn. This is where we're going to pick up today with this process of Megasporogenesis. So Megasporogenesis is a process that produces one. So you go from one functional megaspore, and that functional megaspore will ultimately give rise to the female gametophyte plant that bears an egg. So Megasporogenesis is the process of going from one functional megaspore to female gametophyte plant with egg in angiosperms. Right. Here's a little overview of the process, and we're gonna go through these steps on the subsequent slides. So don't get overwhelmed, but this is a nice little roadmap of where we're going to go in the next few slides. So you have an ovule, which is 2N. Inside of that ovule is a megasporocyte, which is also 2N. That's going to undergo meiosis, 1 and 2, and cytokinesis, the result of which will be one functional megaspore. This is stuff we've kind of already covered in the other, when we first talked about the ovules. Even in the gymnosperms, right? One functional megaspore should be a familiar concept at this point. That one functional megaspore will go through mitosis, and then mitosis, and then mitosis again, and then finally cytokinesis, and then the result is a mature ovule that is 2N that contains a female gametophyte, which is 1N, with an egg, which is 1N. So note that, see here, developing ovule 2N. In all of these steps here, this, then all these steps of mitosis, 3 rounds of mitosis, and then cytokinesis is all happening inside of the ovule. After all these processes finish, you end up with one female gametophyte plant with an egg that are 1N, but notice that the ovule is still 2N, right? Because all of these things are occurring in the ovule. The ovule is part of the sporophyte plant. All right, let's go through this in pictures. So here you go. You got on the left here, this is an ovary wall of a lily. I'll switch colors to blue. Look, we've cut through this ovary wall, and inside here we have lots of ovules. So after Megasporogenesis occurs, inside of each of these ovules is going to be a female gametophyte with an egg in it, right? So there's going to be an egg in each of these ovules after Megasporogenesis happens. So we'll go through the process inside of one of these ovules, right? This is a close-up of ovule. This is a close-up of this ovule here. Okay. Here you go. Developing an ovule and then mature ovule at the end. But inside, this is what's happening. Oh, all right. Developing ovule, mature ovule, and after Megasporogenesis occurs, the result will be this. Look, female gametophyte plant inside of the ovule. But how do we get there? So here's how we get there. Inside, so here's your developing ovule. Notice in these slides that are coming up, in the diagram, you see this like little eye. This little eye is this little eye. See this little M. This little M is this little M, etc. This little C. This little C is this little C. So on this slide and the slides that are coming up, when you see these little letters on the diagrams, I tell you what they are in the text. So inside your developing ovule, you have an integument. One integument at this point because it hasn't grown the second one yet. And inside of that integument, you have the megasporangium. And inside of that megasporangium, you have that megasporocyte, right? All of these things being 2N. Megasporocyte is going to grow, megasporocyte is going to undergo meiosis 1 and 2 and cytokinesis. Remember, when you have meiosis 1 and 2 occur, right here is your megasporocyte. Going to go through this process here, meiosis 1 and 2 plus cytokinesis, such that you end up with a tetrad of spores. Here they are stacked on top of one another. Here's 1, 2, 3, 4. So you have a tetrad, but you're ultimately going to lose 3 of those and keep just 1 of those megaspores to have your functional megaspore. But I'm just pointing out, you still do go through the regular processes of meiosis, right? You don't skip the fact that you end up with a tetrad. You still end up with a tetrad, but 3 of those products will go away and 1 megaspore will stay. Now we have 2 integuments. Here's our Tetrad of Spores. Again, here's our tetrad. Whoops. 4 cells stacked on top of one another. Biggest cell on the bottom is going to be the one that becomes the functional megaspore. This is all occurring inside of the megasporangium. And like I said, yeah, now we have the second layer of integument forming. So 2 layers of integument in B. All right, now 3 of those spores are going to abort, and we're going to keep just 1 megaspore, right? Functional megaspore, which is labeled here FS. And the other spores will just be discarded. All right, now this is that functional megaspore here, right here, functional megaspore. It already has 2 nuclei because it's already gone through mitosis, 1 round of mitosis, but notice, no cytokinesis, no cytokinesis. And this is why back in the beginning of this course, I always made a big deal to say mitosis and cytokinesis, because you can have mitosis occur without cytokinesis. And this is a good example of that here. We're going to have 3 rounds of mitosis occur before cytokinesis ever occurs. So here's the first round of mitosis. Functional megaspore divides into 2 daughter nuclei. Here's our second round of mitosis. Those 2 daughter nuclei are also going to divide such that we now have 4 daughter nuclei total. And this is all occurring inside of those integuments, right? There they are, inside of the megasporangium. And then finally, we're going to go through 1 more round of mitosis. So those 4 daughter nuclei are each going to divide. So we get 8 daughter nuclei total. So 3 rounds of mitosis, and then cytokinesis, and then cytokinesis. And just so you know, let me draw this here. You might notice, here you go, and we have how many? 1, 2, 3, 4, 5, 6, 7, 8, right? 6, 7, 8 cellular products. Aha, that's going to be your female gametophyte plant, right? That's going to be your female gametophyte plant. All 8 of those products inside of that 1 cell. There you go. Now we have the female gametophyte plant. Here are the cells that we know occur right here. I'll do this time it's flipped upside down from the one that we first saw. So your 3 antipodal cells that are labeled AT. Here's your 2 polar nuclei. There's 2 of them in there. PN. 2 synergid cells. Oops. Right here. And oh boy. And right here. And then, of course, probably most importantly, the egg, which is right here in the middle, right there. Female gametophyte plant. Super, super small. Has to fit inside of an ovule, right? And you could have 1 flower that has like 20 ovules. So just imagine how small those female gametophyte plants are. Any questions about Megasporogenesis? No. Okay. All right, so now we're in the mature ovule. Mature ovule has these 2 integuments. Change colors, I guess, green, right? 2 integuments that are enclosed. Oh, actually, I think I color code this for you guys. I think I do, rather than me trying to draw. Let's see. Yeah, boom. Thanks me. Integuments that surround the megasporangium. The megasporangium surrounds the megaspore wall, right? Functional megaspore wall, but then the megaspore wall is going to contain inside of it female gametophyte with egg. So if I add the colors on it, here you go. The 2 layers of integument are in green. The megasporangium is in blue. Megaspore wall is in red. And then the female gametophyte is yellow and the egg is purple. Really loud bird outside. I don't know if you guys can hear it. Can you hear it? Sounds like a laser. Pew, pew, pew. Okay. Right. So the point is the egg is very well protected, right? Well, way better protected than we've seen in all the plants so far. If you compare it back to like the non-vascular plants, I mean, geez, we've really become way, way, way more protected now that we're here. But even compared to the gymnosperms, we're better protected. And so that's how we get to the female gametophyte and the egg. Now let's talk about the male components, right? So male components collectively are called the stamen, or if you want to be more fancy, you can say androsium, that's fine too. And the stamens consist of the anthers and filaments. The anthers, oh, so here's the stamen. Like everything inside the red is a stamens. The anthers are the thing at the top of the stamen, and that's where pollen is going to be produced. And the filament is just a stalk that holds the anther out of the flower. Right. And so we talked about where the female gametophyte is. Now we'll talk about the male gametophyte. So we should already know from our lectures in the gymnosperms that the male gametophyte plant now in the seed plants is this pollen grain, I've already talked about that a bunch of times. And it's basically the same here in angiosperms. The only thing is now the pollen grain is even more reduced than what we saw in the gymnosperms, because now it is only 2 cells inside of that megaspore wall. So we have lost the prothalial cells. The prothalial cells are gone. Remember when we were covering the gymnosperms, sometimes there was 1 prothalial cell, sometimes there was 2 prothalial cells. In either case, we didn't know what they did. We don't know what they do. By the time you get to the angiosperms, it's lost entirely. So that's why we think the prothalial cells may be kind of a vestigial thing. Because in the gymnosperms, we don't know what they do, and then in the angiosperms, they're lost entirely. They're gone. So now the male gametophyte plant is reduced to only 2 cells inside of this megaspore wall: pollen tube cell, which still grows pollen tube, generative cell, which still generates 2 sperm. That has not changed. So we had Megasporogenesis, where you go from a Megasporocyte to a female gametophyte plant, right? Megasporocyte to a functional megaspore to a female gametophyte plant. That was Megasporogenesis. Now we'll do Microsporogenesis, which is the process of producing microspores, which will ultimately give rise to the pollen grains. And the pollen grains contain the now very, very, very reduced male gametophyte plant that only consists of 2 cells: pollen tube cell and generative cell. 2 cells. Mind-blowing. I mean, like, think back to the non-vascular plants where we had entire male gametophyte plants. We had entire female gametophyte plants like with rhizoids and other structures growing off of it, and lots of antheridia and lots of archegonia and just a whole organism. And now the male gametophyte plant is just 2 cells. 2 cells. Pretty incredible. Still gets the job done though. All right, so here we go. So some of this is going to be a review with Microsporogenesis. Remember also, just so I know the words can seem kind of scary, but remember, Genesis means the beginning of, and then microspore refers to microspore. So it's the beginning, the word itself means beginning of microspore, whereas Megasporogenesis means the beginning of megaspore, right? Just so you know. But anyways, here you go, Microsporogenesis overview. You have a microsporangium, aka pollen sac, which is 2N. Inside of that pollen sac, aka microsporangium, are microsporocytes that are 2N. Microsporocytes are going to undergo meiosis 1 and 2 and cytokinesis to produce microspores. Microspores are going to undergo 1 round of mitosis and cytokinesis, and the result is our pollen grain, aka our male gametophyte, that consists of only 2 cells, the pollen tube cell and the generative cell. Okay. So where is this all happening? This is all happening inside of the anther. So the anther is where those microsporangia, aka pollen sacs, are located, right? So on the left here, you have a cross section, or you have a picture of a lily. I guess I'll use blue here, right? In the center here, you see the style of the pistil and then the stigma of the pistil right there. But then surrounding it, we have 6 stamens. Here's 1 stamen, here's another stamen, here's another one. And each stamen is made up of a anther, which is this top reddish part, and the filament, which is the stalk that holds it up. If you were to do a cross section through one of these anthers, that's what you see here on the right. And you could see each of these things. So this is 1 microsporangium, aka pollen sac. This is another microsporangium, aka pollen sac. There's a total of 4. And if you do a cross section through it, kind of almost looks like a little butterfly thing, which we'll see on the next slide. So here you go. Here's that butterfly. It's a cross section through an anther, right? What do I want to say here? Right. So each, I think I actually draw it myself on here. So you have 4 microsporangia shown here in this cross section. Inside of each microsporangium is microsporocytes that are 2N. So here's our 4 microsporangia in red. So microsporocytes are inside of there. Each microsporocyte, which is 2N, is going to go through meiosis and cytokinesis, which will make a tetrad of microspores. The microspores will eventually break up, right? The tetrad of microspores will break up. So you have individual microspores. There's 1. And then that microspore is going to undergo 1 round of mitosis and cytokinesis and become a pollen grain, which is a structure now that only consists of 2 cells, pollen tube cell and generative cell, and is effectively the male gametophyte. And the pollen grain is what is going to get released from the anthers. That's what's going to be released. Microspore is not going to be released. Microspore stayed inside of the anther of the microsporangia of the anther to become pollen grain. So microspore doesn't get released, pollen grain gets released, just like in the gymnosperm. Here you go. Eventually the microsporangia, aka pollen sacs, will open and they will release the pollen grains out. Okay. Now we know where the egg is and the female gametophyte. We know where that is. That's in the ovule. And we know how pollen grain, aka male gametophyte plants, are made. So the only thing that has to happen now is they have to come together, right? So for angiosperm reproduction to occur, pollen grain and sperm and egg need to fuse, right? All right, so let's do that. And that's going to happen via something called pollination. So here you have an anther. Sorry, here you have a stamen. This whole thing is a stamen, right? And at the top of that stamen is an anther. Microsporogenesis already occurred, and now pollen grain gets released. There it goes. Wee, goodbye. I'm out of here. Fly away. Well, no, not necessarily fly away. Get released, right? It could just be on the outside of the anther. Get released. And then via something we call an agent of pollination, right? Agent of pollination, the pollen grains are going to be taken to the top of a stigma of a flower. So that's what you see here. Pollen grain, whoop, go into the top of a stigma. When pollen lands on the stigma, so the transfer of pollen from an anther to a stigma is the process of pollination. Remember, in the gymnosperms, we had when the pollen grain would land on the sticky drop of the ovule, right? That was pollination in the gymnosperms. In the angiosperms, pollination is when the pollen grain leaves the anther and lands on the stigma. That's pollination in the angiosperms. Here, this is a great microscope slide showing you this is the top of a stigma. This stigma happens to be branched into 5 pieces. So Brittany, to your question last time about like, I think of the pistil as a single unit. Yes, a pistil is a single unit, but there can be components, right? So for example, if we're talking about flower parts, right? 4s and 5s versus in 3s, this stigma has 1, 2, 3, 4, 5 branches. So do we think, what kind of plant do we think this stigma must be attached to? There's 5 branches on this stigma. Yeah, it's a eudicot, perfect. So that's what I was trying to get at last time, but we didn't have good slides to really show that. Right. If this stigma had 3 branches on it, for example, then we would think it was a monocot. And each of these branches is actually going to lead down to separate compartments where ovules are located, just so you know. Anyways, great microscope slide showing you pollen grains on the top of a stigma. So the pollen grains are those little yellow balls. There's tons of ways that pollen grains can get from the anther to the stigma. No matter what way it is, it's still called an agent of pollination. So you have abiotic. A means without. A means without. Abiotic, without biotic. Without biotic, abiotic. So sometimes the pollen gets brought to the stigma via water and wind, right? Not living organisms, but water and wind. But then you also have biotic agents of pollination, insects, birds, reptiles, small mammals, including bats. Here is an example of pollination via an abiotic agent, right? Wind on the left, which if you have allergies, you are annoyed at that. And water on the right. So all that stuff is pollen. Here's examples of some insect agents of pollination, right? You got bees, of course, but also butterflies and moths and even wasps. Flies can also be agents of pollination. Look, beetles could even be agents of pollination. You don't think of beetles. You always think of bee, right? Just bee, right? Birds and bees. You always think of bee. And by the way, look at all this pollen this bee is packing. Look at this. Look at all that pollen stuck to that bee. Anyways, yeah, but it's not just bees and wasps and butterflies and moths. It's also beetles, right? Look at the beetle on the left, covered in pollen. Hummingbirds. Reptiles, look at this guy. Aw, right? Reptiles can be agents of pollination. Bats and other small mammals. I love this slide. I love this slide. Oh my gosh. I just love this slide so much. I mean, look at this guy. Look, I am covered in pollen. Having a good day. Yeah, bats are usually agents of pollination for flowers that open at night, right? So you can see here, nope, they're not allergic. They're not allergic, Brittany. So cute. Yes, I love this slide. But then also look at these two, these little mousy things. Aw. All right. Sorry, even plant people like seeing cute little animals. Yeah, so mammals can also be agents of pollination. Either way, no matter how it happens, you have pollination occur and the pollen grain lands on top of the stigma. All right, that has happened now. Now that the pollen grain's at the top of the stigma, what's going to happen? Pollen grain, right, landed on top of the stigma, and it has in it this tube cell and the generative cell. So once the pollen grain lands on the top of the stigma, chemical interactions occur between the top of the stigma and the pollen grain. And the pollen grain is going to germinate and absorb water from the surface of the stigma. And the pollen tube cell is going to grow down the style. I guess I'll use black here. So here you go. Here's the top of the stigma right here. Here's a pollen grain at the top of that, right? Pollen grain is going to germinate and then grow pollen tubes down the style of the pistil. Here you go. This is a pollen grain that has germinated. And the pollen tube cell is, you can see very clearly here. As this pollen tube cell grows down the style, the generative cell is going to divide via mitosis and produce 2 sperm cells. So here you go. Here are the nuclei of those 2 sperm cells. This is the nuclei of the pollen tube cell. This is the, but remember, this whole thing is pollen tube. It's just, it has to have a nuclei in this in order to keep elongating. But yes, the generative cell is going to produce 2 sperm cells that are not motile, right? They do not swim. These do not swim. Instead, as the pollen tube cell grows down the style, the 2 sperm cells are brought with it as it grows down, So they don't swim anymore. Alrighty, so pollen tube cell is going to grow until it finally reaches an ovule. So that's occurred here. Look, so here we go. Pollen tube cell growing. There's a sperm. I'm going to go over them and then boom, whoops. Boom, right? Pollen tube cell reaches an ovule. And again, Brittany, back to your thing. Remember, this is a lily, and look how many compartments there are here, right? 1, 2, sorry, it's 6. It's 6. You can see 1, 2, 3, 4, 5, 6. Right, lily. Anyways, yes. So pollen tube cell is going to grow until it reaches an ovule. The pollen tube is going to enter at that hole called the micropyle that's in the ovule that we've learned about before, right? Here's that hole right there. And then once the pollen tube cell grows and reaches an ovule, that's going to allow the 2 sperm cells to be released into the ovule. So this is a close-up of that process inside of that ovule. You can see the pollen tube cell is entering. Oh, sorry. I messed up before. This is not the micropyle. No, Rebecca. Micropyle is at the top of each ovule. So this hole here is the micropyle. Each ovule has a micropyle. Sorry about that. I got a little carried away. So pollen tube cell is going to enter the micropyle. The 2 sperm are going to be able to reach the inside of the ovule and fuse with this. Well, give it a second. Gets a little complicated right here, but pollen tube cell arrives at the ovule. The 2 sperm cell arrive as well. And those two sperm cells are going to meet these things, including the egg and the two polar nuclei. So we have a new thing going on here called double fertilization. Double fertilization. See? Double fertilization. Double fertilization. What? Double? Yes. So you might remember back in the gymnosperms, there were also two sperm cells. One of them would reach the egg and then the other one would get discarded, right? That's wasteful. That's super wasteful. So in the angiosperms, we now take advantage of both sperm. So one sperm is going to fuse with the egg, and that's going to give us our 2N zygote. The other sperm is going to fuse with the two polar nuclei that are each 1N and give us this 3N or triploid product called endosperm. So what is double fertilization? Double fertilization is the process by which there's two sperm, one sperm fuses with egg to make a 2N zygote, one sperm fuses with the polar nuclei to make 3N endosperm. And then you have a seed. So now you have a seed, right? 2N zygote and 3N endosperm is the seed of angiosperm. So here's what I was just saying on the previous, right? Double fertilization, two fertilization events inside the female gametophyte plant, two sperm cells, one sperm cell fuses with an egg to make a 2N zygote, which will grow into the new sporophyte plant, and one sperm fuses with the two polar nuclei to make the 3N endosperm, which serves as a food source for that growing zygote. Right, I already said that. All right, you've probably eaten fruit before. But you may not have realized maybe, and you're going to be like, how did I never know this? But yeah, so every time you eat a fruit, it originally was a flower, guys. It originally was a flower. So here's an apple flower in A. You have Megasporogenesis and Microsporogenesis occur such that you have male and female gametophyte plants, or female and male gametophyte plants, which are the mature ovule and the pollen grain, right? Pollination occurs, double fertilization occurs, and ultimately you end up with seeds inside of a fruit or an apple. So anytime you've eaten a fruit, that was a flower. Here's another example. In blackberry, right? Here's a blackberry flower growing at the top row. Here's what it looks like once it's more like a flowery look, right? Stigma in the middle, stamens all around. But eventually eventually pollination occurs and then double fertilization, and then you end up with a blackberry. There you go. Delicious. Brittany said, thank you. This is what I needed to see. Yes, we'll see even more. We'll see even more. In fact, we'll see some right now. I'm gonna see, we could watch this. I think I have to share this with you guys. 1 second, pause, stop share, and then share this here. Make it big. Hopefully you guys can see the video. So this is a time lapse. This is a pear flower. Here's pear flower opening. Pear flower opening. There you go. Look in there. Green stuff is the top of the stigma. Purple things are the stamens, right? Now the anthers of the stamens are going to open. See that yellow stuff? That's pollen. Eventually the pollen from those anthers is going to get brought to the top of the stigma, when that will be pollination. Then the pollen tube cell is going to grow down, meet the ovule. The sperm is going to fuse with the egg and the two polar nuclei. And here you go, look, a pear. A pear. This is how we get fruit. Look at that. There's a pear. Still growing, but you get the idea. Now I'll post all these videos on Blackboard as well. So you could watch them again at your convenience. And then I have another one here. Oh boy, hold on. I have another one here. Yes, here, and I'll share this with you. Oh boy. 1 second. Do this here. All right, and then I will share this with you guys. Share screen. Here we go. Hold on, there's an ad. Shh, quiet ad. All right, skip ad. Here we go. This is a blackberry fruit to a flower. So here you go, blackberry flower. Sorry, blackberry flower to fruit. Blackberry flower. In there, the greenish thing is the tops of the stigma. The petals now have fallen off. You have the stamens surrounding the stigma. Pollen grains are going to land on top of the stigma. Pollen tube is going to grow down. Egg and sperm will get fertilized, and also the double fertilization, right? But look, blackberry fruit from a flower. From a flower. Yeah, so again, I'll post all these on Blackboard as well, so you guys can watch them at your convenience. And let me share this and get big. I had a question. Yeah, yeah, I'm just now seeing the chat box. Wow, yeah, it is cool, right? And yeah, yeah, all those little like things around it are the stigmas. Yes, correct. I thought you guys would like this. All right, so here you go. So when we last left off on this diagrams, right, we had a finished seed, right, which inside of the seed had a 2N zygote and the endosperm. So this slide just summarizes everything we've just talked about, today, right? This is the male part, the stamen. Pollen grain is going to get released. Eventually, it's going to land on top of the stigma, aka pollination. Eventually, a pollen tube cell is going to grow down. It's going to meet with an ovule. It's going to allow the two sperm. Look, here they are. They're traveling down. Boop, 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 boop. Right. To go into the ovule and fuse. One sperm will fuse with the egg. One sperm will fuse with the two polar nuclei in the process called double fertilization. Result is a 2N zygote and an endosperm, which is 2N zygote, 3N endosperm. Oops. And that 2N zygote will grow into your embryo. The embryo will consume the endosperm because the endosperm serves as food for the developing embryo. And the integument will turn into a seed coat. And the extra credit lecture on Monday will talk more about that process. And then eventually the embryo will grow. Eventually the embryo will be able to leave the seed by growing roots and growing shoots, and then the seed eventually will just go away, right? Because it doesn't need it anymore. Okay, what are we doing here? Good. Here you go. This is a seedling emerging from the seed. These are the part we've learned about parts of these embryos before, but just this is the root emerging from the seed. Usually the root emerges first because the embryo has food in the seed via the endosperm, but the embryo does not have any water in there. So normally when the embryo does emerge, the first thing to come out is roots because it needs water. And then once it has water, it'll grow a shoot system to start making its own food. But water is the most important thing because there's no water inside the seed. All right, here you go. This is growth of the new sporophyte plant. So here's your seed over here. First thing's going to emerge is this root system so that the new sporophyte plant can get water. Here's that term we came across last time, cotyledons. So those are the embryonic leaves. You can see them right here. There's two there. There's two there. Yeah, so this is just showing you the progression of a embryo growing into a new sporophyte plant. New sporophyte plant. But look, eventually the cotyledons are not needed anymore, and they kind of wither and dry up once the plant has its own mature leaves. Just so you know, these words hypocotyl and epicotyl, they just refer to parts of the stem. Hypo means below, and the cotyl part refers to cotyledon. So hypocotyl is the stem below the cotyledons. Epicotyl is the stem above the cotyledons. Yep, this is how a seed grows. And actually, we're going to watch videos of it as well. So here we go. Oh, no, that was this plant here, this garden bean, has two cotyledons. And it has leaves. Look at the leaves. Look at the mature leaves. Net-like venation. Two cotyledons. So what type of plant do we think this is? It has net-like venations. It has two cotyledons. What must this? It's a eudicot. Thank you, Ben. Yes. Whereas here, we only have one cotyledon, although it's not labeled. Don't worry about that. The leaves have parallel venation. So in this example, this is a monocot here. All right, now we're going to watch a few of these. Let me pull this. Oh my gosh. All right. I'm going to stop sharing that. And I'm going to share this. We pause and go back to you guys here. Share screen. Okay, hopefully you guys can see. Oh, there's an ad. Sorry. Don't listen to this lady. She doesn't know what she's talking about. Don't listen to her. Okay. Here we go. So this is a sunflower, from a sunflower seed to a flower. So look, here we got the seedling emerging. Those round things there, here, pause real quick. Yeah, don't listen to that lady. She don't know what she's talking about. These round things here that just emerged, like here's one, the other one's on the other side, you can't see it, are those cotyledons. So the cotyledons are the first leaves to emerge. Here we go, gonna keep growing here. Now we have the mature leaves. So this is a time lapse of, you know, this plant growing over time. Lots of different photos. Yep, starting to grow. Now look, the cotyledons are withering. They're going to fall off. It doesn't need the cotyledons anymore. So they're going to wither. And it's growing more and more and more leaves from the apical meristem, right? So the apical meristem is here at the top, growing all that stem tissue, growing all that leaf tissue. It's growing all that stuff. It's lengthening. Now we see a flower bud emerging from the top. Here we go, flower. Now we're close up on that flower bud. Come on, really taking its sweet time here. All right, so flower bud starting to open. Remember, this is a sunflower, or a sun inflorescence, I should say. Here we go. starts to open. You can see all the disc flowers are exposing their stigmas. You can see the stigmas coming out. And then there you go. Sunflower, right? Sun inflorescence. Eventually all of those disc flowers in the middle will become pollinated and turn into sunflower seeds. All right. Next, we got this one here. All righty, let me make it big. All right, this is a kidney bean. So look, here's kidney bean in the dirt. First thing to emerge is the root. So that is a root emerging. Look at that, via apical meristematic growth, apical meristematic growth. Now the secondary shoots are, secondary roots are starting to come off of that first root. And you can see the stem elongating, that's that green thing. Stem is elongating. Now the two cotyledons are going to, so you see the mature leaves kind of poking out. Those round things though that look like peanuts are those cotyledons. See the growth, primary growth occurring at the apical meristem, which is at the tip. Got two mature leaves at this point. Cotyledons are starting to wither away. Growth continuing, primary growth continuing, more and more leaves being made at the top here. Look at that root system though on the left, got really, really developed. More growth, more growth, more growth, right? Pretty cool. Isn't that incredible, guys? I mean, you go from a seed to a whole plant. It's just incredible. All right. And then the rest of this video shows you the roots growing. So you can watch that on your own if you'd like. And then, okay, this one is 8 minutes long, I think. So we're not going to watch all, oh, hold on. We're not going to watch all of this, but I just want you guys to see, this is a really cool, I just found this video this year. And this is 8 different, oh, don't listen to them. Nope, be quiet. We don't care. Go away. This is 8 different seeds growing into 8 different plants. So it's really super cool. Again, I think this is like 8 minutes long. Yeah. So we're not going to watch all of it. I will post the video on YouTube, but look, so there's basil seeds and then whammo, basil plants growing. Yep. Here. Yeah. Right. Basil. Here, look here, they're planting a cranberry bean. A little water. And you can do this at home, guys. Like you can experiment with things like this and watch plants grow on your own. Here, the one I wanted to show you guys here, I think it's this. Oh, this sunflower again. Oh yeah, this is cool. This is cool. This is a pepper, right? So they just took a seed out of a pepper, put it in the dirt, give it a little water, and then you gotta wait. Ah, and there, boom, there you go. So here's our pepper plant that's coming. Pepper plant. I love this video. I'm definitely gonna use it from now on. It's super cool. Like I said, there's 8 different types of plants. So I'll post the videos on YouTube or on Blackboard. This is our pepper plant, growing, 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 growing. And then eventually, gonna make some flowers. So here's flowers on the pepper plant. Pollination is going to occur, double fertilization is going to occur, and then what do you know? Bell peppers, which is a fruit, Yeah, so I will post those videos on Blackboard. My mom planted lots of peppers in the summer. Yeah, they're fun to grow. super fun to grow. All right, I'm going to share screen. All right, we got about 20 minutes left, I think. My mom does too. You know, my dad grows peppers, and in the winter time, because he just can't say goodbye to them, he brings them all inside, and they've actually made peppers over the winter a few times. Drives my mom nuts because it's just like all these pepper plants from outside come in and it crowds up everything. All right. Yep, from one tiny seed. So we've covered the angiosperms. That's basically all of the lecture material that we have for the angiosperms. So before I have another video we're going to watch very quickly, but before we do watch that, I just wanted to ask if there's any questions about anything that we've covered, or anything with the angiosperms. I know we only really finished it, you know, this week, so you might need time to like look at the slides and think about it a little bit and then ask me questions. But if anything comes to mind right now that you're wondering, let me know. Let me know. So I have a resource for you. So while you think about that, we do have special guests next week. So we're going to learn next week from real life people about different careers you can have with plants. Hold on. Different careers you could have with plants. But so you know, I've also posted this. Let me share this with you guys. I will also included in this PowerPoint is this link, which kind of walks you through like different jobs you could have working with plants. So it's kind of cool. You can select a degree, you can select a category, select a subject. And then it kind of gives you a roadmap of what types of jobs you could have, depending on what things about plants that you like, right? So you could, if you like horticulture, you can be an arborist, or a forestry consultant, or environmental scientist, bioprocessing engineer. Within forestry, you have forestry scientist, forestry technician, horticultural scientist, irrigation engineer. There's overlap there between that and agriculture. Logging crew member, plant geneticist, soil scientist, and there's a whole bunch of other things here too for your convenience. So that's a very useful useful website. Yeah, GMO work seems so fun. Yeah, I mean, there's a lot of different types of opportunities in so many different sectors of plants and plant science, but also like you don't need a degree in plants to work with plants. Like even before I had my degrees, you can work doing garden work, internships. There's all sorts of stuff. You could even volunteer at a community garden, things like that. Let's see. So but next week we'll have a lot of great. Okay, we're doing great with time. Next week we will have special guests who can really tell us what their life is like while they're working with the plants in the ways that they work with plants. All right, but in case in case you want more resources, this plant-based career website is good. So just to, next week we're not having lecture material, right? So we're having our special guest. So I just want you guys to keep in mind as you prep for the final exam, you really want to keep in mind those core course concepts, which are outlined on the syllabus in case you don't remember what they are. But yeah, the final exam covers angiosperms and then the core course concepts. In particular, going from a spore shedding to a seed shedding plant and understanding how the spore bearing structures have become more complex over time. So that's a core course concept. No, you don't need to know everything about, lycopods and cycads, but you do need to understand how like things like angiosperms and conifers, but mostly angiosperms are more complex than these other things. than these other things. So when I say spore-bearing structures, I'm referring to sporophytes and sporangia. Sporophytes and sporangia. So that's something you should keep in mind as you study. Another thing is understanding how the spore-bearing structures have become more complex over time. Understanding how the gametophyte have become more reduced over time. This is a shift, right? We saw this shift in dominant generation. That was important if we were going to go from a very large gametophyte plant to a very small gametophyte plant. We have exosporic versus endosporic gametophytes. We have homospory versus heterospory, etc. So understanding how the gametophytes have become more reduced over time. All right. Last thing we're going to do today, we're going to watch this video, which I always do in this class because it's a great video to end on. And let me go here and here. I'll wait for the ad to finish so I don't have to yell at the ad for you guys. Skip ad. All right, hold on. I got to share with you guys. This time I'm going to share it with the sound of the video. So if I want to point anything out to you guys, I'll write it in the chat box. So I'm going to share sound. And I will share this. If I'm going to play it with sound, if it's too, I'm gonna turn the volume up to begin with, but if it's too quiet, let me know. But I don't want to blow your guys' mind. Whoa.
It's great being here at TED. You know, I think there might be some presentations that'll go over my head, but the most amazing concepts are the ones that go right under my feet. The little things in life, sometimes that we forget about, like pollination, that we take for granted. And you can't tell the story about pollinators, bees, bats, hummingbirds, butterflies, without telling the story about the invention of flowers and how they co-evolved over 50 million years. I've been filming time-lapse flowers 24 hours a day, 7 days a week for over 35 years. To watch them move is a dance I'm never going to get tired of. It fills me with wonder and it opens my heart. Beauty and seduction, I believe, is nature's tool for survival because we will protect what we fall in love with. Their relationship is a love story that feeds the earth. It reminds us that we are a part of nature and we're not separate from it. When I heard about the vanishing bees colony collapse disorder, it motivated me to take action. We depend on pollinators for over a third of the fruits and vegetables we eat. And many scientists believe it's the most serious issue facing mankind. It's like the canary in the coal mine. If they disappear, so do we. It reminds us that we are a part of nature and we need to take care of it. What motivated me to film their behavior was something that I asked my scientific advisors: what motivates the pollinators? Well, their answer was, it's all about risk and reward. And like a wide-eyed kid, I'd say, why is that? And they say, well, because they want to survive. And I go, why? Well, in order to reproduce. Well, why? And I thought that they'd probably say, well, it's all about sex. And Chip Taylor, our monarch butterfly expert, he replied, nothing lasts forever. Everything in the universe wears out. And that blew my mind, because I realized that nature had invented reproduction as a mechanism for life to move forward, as a life force that passes right through us and makes us a link in the evolution of life. Rarely seen by the naked eye, this intersection between the animal world and the plant world is truly a magic moment. It's the mystical moment where life regenerates itself over and over again. So here is some nectar from my film. I hope you'll drink, tweet, and plant some seeds in the pollinator-friendly garden. And always take time to smell the flowers and let it fill you with beauty and rediscover that sense of wonder. Here are some images from the film. Thank you. Thank you very much.
All right, hold on, let me close this. Yay, okay, so hopefully you guys enjoyed that. Yeah, let's see if I have anything else on this. All right, let me share. Yeah, I'm sharing this, make it big. Yeah, so I'll post all these videos on Blackboard in case you have connection issues or anything like that. Just a few other things. So we have an extra credit seminar on Monday. So please attend that if you want to get extra points toward the final exam. Next week, we're doing our special guest, which I'm so excited about. This is the first time I'm ever doing a special guest thing, obviously, because the virtual format allows me to do this. So I'm very excited. I hope you guys are too. We have the phenology assignment is due on the 29th. You guys have your lab exam on the 30th. For the final exam for lecture, I'm going to upload a study guide around the 30th. I won't do it before that because you guys will be focusing on other exams and the lab exam anyways. And then, yes, there's a course survey that I sent out. So pretty please fill out the course survey and be honest on it so that I can adjust. Hopefully I do get to teach this class again, which is a big unknown, but even if I don't do it at Rutgers, maybe I'll be able to teach it somewhere else because I really, really love this class. But yes, please fill out the course survey so I can improve upon my class. And then just a reminder that the final exam is on May 11th with a 10 a.m., not 8, whatever it says, a 10 a.m. start time, right? And this is part of the slides. So the QR code is here to fill out the course survey in case you guys like to do that. All right, stop sharing that. All right, guys. Well, I hope you enjoyed learning about plants, and I'm looking forward, like I said, to hearing from our special guests next week to see about how different jobs you can have working with plants and the experiences that people have had working with plants. So I'm very excited about that. And if you guys have any questions, I'm happy to answer them here or in a follow-up email. But I just want to say, and I'll say this again next time, next couple of times, but I really have enjoyed having you guys. I know the virtual format is weird, but I've done my best to have it be as good as it can be, considering I can't actually make you hold leaves or make you smell things. But I hopefully you guys have learned a lot. I think many of you came into this class probably being mad that you had to learn about plants. Hopefully now you're a little less mad or maybe even excited about plants. Hope your phenology assignments went well. Hope you got to see some plants flowering over the course of your observation period. But that's all I have for you guys today. If you guys have any questions, feel free to write it in the chat or shout it out. Otherwise, have a great day. And thank you guys so much. Yep, have a great weekend, guys. Have a good lab first and then have a great weekend.
Professor.
Yes.
Do you know why a lot of people are allergic to pollen, even though, we were talking about it and it seems like it's very simple, like 2 little cells flying around.
Right.
But mad people are allergic. So it's like, do you know why?
Well, so in terms of, I don't know. I'm not an expert in like allergies or anything like that. If I had to guess as an evolutionary biologist, I would say that probably most plants, so think about evolutionary time and think about humans, and like we didn't used to be able to go all over the world, right? We used to be pretty limited in general to like where we went, right? I'm talking like before civilization was even really built, but even after civilization was kind of built, we still didn't really move around that much. And so a given group of humans would be exposed to certain plants that would be occurring like around them a lot, right? Like you would become accustomed to those kind of pollens. But now that humans, we move all over the world and we put plants in places from all over the world, right? Like if you walk through Journal Square, or if you walk through, Manhattan, you're going to be walking by plants that are from all over the world, usually Europe, but and Asia, but still, I mean, the point is that I think part of the reason people are allergic is because now we've really moved plants and people all over the world. And so we're exposed to types of pollen that maybe we hadn't been so much in the past. There's other reasons, I'm sure. It could also, I'm not an allergy specialist, but it could also be, I know me, for example, I grew up, when I was a little kid, I lived in New York City, and then I moved to Florida. And in Florida, excuse me, in Florida, things don't bloom all at once. Like in Florida, it's hot, so things bloom kind of like all the time, but like, you know, one day it'll be like that one thing over there is blooming, and then another day it's like, oh, over there that thing is blooming. But it's not like spring in the Northeast where it's literally like cold winter, and then all of a sudden, whammo, everything's in bloom, right? So when I moved back to New York from Florida, I had never had allergies, but then in the springtime, when every single thing in the, it seems like on the planet is in flower, it's also just the load of pollen. Like, that's not always the case. Like I said, like if you live in the tropics, or if you live in Florida or other warm tropic kind of like warm environments, it's not like everything's in bloom all at once. So I think that could play a part in it too, in that a lot of us live in kind of these temperate environments where everything flowers at once. And so even if you aren't allergic normally, it could just be like pollen overload, pollen overload. So again, I'm not a pollen or allergy expert, but that would be my guess. Yeah, it has to do with exposure over evolutionary time or lack thereof, and then just like sheer amount of pollen during some times of year.
Oh, cool. Because for a second I was like, damn, are we allergic to that one cell?
Oh, so that's another thing I should mention. So even though in angiosperms, the pollen grain is that, you know, it's that the generative cell and the pollen tube cell inside of that, you know, cell wall. So even though it's actually pollen tube cell and then like generative cell inside. And yeah, so even though that's the case in terms of like the structure, all plants, so pollen grains from different species have different enzymes on them for many reasons. One reason is because, let's say pollen grain from an apple flower lands on the stigma of a pepper flower, right? And so like if a apple pollen lands on a pepper flower stigma, it's not good for that to germinate, because, and it won't often germinate. I mean, sometimes there's exceptions. Plants have exceptions all the time, but part of the reason that germination won't occur of that pollen grain is the enzymes that are on the pollen grain and the enzymes that are on the stigma. There's a lot of chemical interaction going on that we don't really talk about in this class. And so that's why certain types of pollen might make you feel allergic, whereas other types of pollen won't. And it has to do with the chemical nature of those pollen grains. Yeah. Good questions. Yeah. All right. Well, if anyone has any other questions, happy to answer them. Otherwise, have a great one, guys. I'll talk to you guys later. Bye.